This material is made available to you by or on behalf of the University of Melbourne under Section 113P of the Copyright Act 1968. It may be subject to copyright. For more information, visit the University Copyright website.
What's been going on? Did you mind let's keep promotion up last week? Very boring. Very boring. I took your advice and provided ChatGPT. And Ella provided some very, very nice updated metrics.
Drop those in ChatGPT the last
Exactly. We can call them a lot of.
You gotta edit it.
I did a little bit, and I also I looked at lots of other people's apps in the years past, and I was like, hey, listen, guys. Now I'm asking them to look at this, please, and just give some feedback on that. So it was just like make it consistent. So that's done, and it feels good. Yeah. Kim's too busy to write the heads report, so I think the heads report. Yeah. Which has happened every year. Not that have you had a chance to speak much to Kristen? Yeah. I spoke to her briefly about the hub last week. And yeah, she seemed very enthusiastic about build up more clinical research. She was, I think, because she's been doing the hotspot thing for a long time. I spoke about how to really trademark the skills to
I need to actually.
You said you know, I've been watching her first.
Oh, no, Portugal is worse. Okay, yeah. That's good. I was just excited at the mother. Yeah. Well, exactly. I mean, it is, right? Well, we just got our feedback like yesterday.
Okay.
I think that's something because Hi everyone, we're gonna get started. So today we have a very special speaker from Psychiatry. He's just joined in February. I'll do the introduction in a minute, but first I just want to acknowledge that we are on the lands of the Wurundjeri people. I pay my respects for the elders past and present. And I acknowledge that the Indigenous peoples have always been making discoveries and sharing knowledge much before these buildings of the University of Melbourne were built, and we are so lucky to continue that legacy. So, Professor Josselin Houenou, I can't say his last name. I practiced and still got it wrong. Can you say it?
Yeah, that's okay.
It's okay. Okay. I'll just go by Josselin. I think that one I can say. So as I said, he just joined in January, all the way from France. He is a professor of psychiatry at the Northern Health in Department of Psychiatry here at the University of Melbourne. He's done his medical studies in France, in Paris, as well as his PhD. And he was also a visiting scholar in Montreal, in Canada. His lab was at Neurospin in France, which is a really famous neuroimaging platform. And his lab was there from 2013 to 2025, so just really very new to our university. His main focus is bipolar disorder, which I think that's what we will hear mostly about today, but also schizophrenia and autism using structural MRI. So thank you so much. Just looking forward to your talk.
Thank you. Thank you. I will speak about bipolar disorders, mostly about what I've done and what is my project for the next 10 years. So first, the place where I come from, you may have heard it recently. This is called Neurospin. This is a neuroimaging platform, mostly dedicated to ultra-high field. This was all built around the project of 11.7 Tesla human scanner that just produced its first images of a living brain last year. You may have seen them, like 200 microns of resolution. Is this very good? And it's one special thing that may have helped, that has helped me much, is that it's located in the French Atomic Energy Commission, which may seem weird, but it's because it's mostly a physics facility, not something else. Anyway, it's led by someone you may know, like Stanislas Dehaene. Easier to say. So it's at the interface between clinical studies and physics. So I will speak mostly about bipolar disorder. I think most of you are familiar with bipolar disorder, but just to give you a glimpse of it for those who are not, this is a major chronic mood disorder in adults, affecting 0.5, 1% of the population. There are some specific things. First, its prevalence is the same all over the world. We do not know why, but it's really quite stable all over the world. The sex ratio, because sex ratio is quite specific to bipolar disorder, is 1. It affects equally men and women, which is not the case for any anxiety or mood disorders, which affects mostly more women than men, except for OCD, which is on the contrary. But this is really specific to have a mood disorder with a 1-to-1 ratio. We do not know also why. So we do not know much, so it's easy to do research in this field. It has been conceptualized by Emil Kraepelin more than 100 years ago. One interesting thing is that you can have access in the internet to his book, which is called in English Manic Depressive Illness. And when you look at it, it's quite a very good description of what we know today. So we have to make some progress. It has been conceptualized at this time, but we can find in the literature, like in the ancient literature, in Egypt, Rome or Greece, descriptions of really what looks like bipolar disorder. So there are a few things in French still. Patients suffering from bipolar disorder will usually have an onset of their disease between 15 and 25. It can be later, but the most usual is between 15 and 25. And they will experience either depressive episodes, severe depressive episodes, or manic episodes. For those who have not seen patients with manic episodes, this is quite easy. This is the contrary, the opposite of depressive episode, with euphoria, psychomotor activation, insomnia, things like this. And another phase of the disease is what we call euthymia. People are out of a major mood episode. They have some sub-threshold symptoms. We will speak about this later, but they are out of an episode, and for the DSM, they are out of everything, like this. just to give you a glimpse of the severity of the disease, out of 100 subjects, patients who will be diagnosed with bipolar disorder, 15 will eventually die by suicide. So these are severe mood episodes, and they have other comorbidities, complications, etc. But just this is the psychiatric condition with the highest prevalence of suicide after anorexia nervosa. But this is really huge. So I was using mostly MRI to study bipolar disorder. And you probably all know these models of emotion regulation. So, because first we focused on emotion regulation as this is a mood disorder. These are the models drawn from the animal literature. Josselin Houenou work on the importance of the amygdala 30, 40, 35 years ago. And then by Mary Phillips in London and Pittsburgh. And current models, which are quite robust now, suggest that when we are doing emotion regulation, so it may encompass things like emotion identification, emotion processing, depends on the studies, we are using two broad networks of emotion regulation. The automatic emotion regulation network, which is quite similar to the old limbic network with gray nuclei, amygdala, hippocampus, which activates quite fast after an emotional stimulus, like 50 milliseconds, before you are conscious of it. You can see in many studies, or intracranial studies, things like this, activation of the gray nuclei and the amygdala. And then, after a few hundreds milliseconds, you will have activation of the blue, here depicted network, which is called the voluntary emotion regulation network with the dorsal cingulate, prefrontal cortices, which comes to modulate the activity of the amygdala through direct connections from the prefrontal cortices to the amygdala and the gray nuclei. There are known tracts linking those two systems, like the uncinate fasciculus, that were already known in the anatomical literature. So this is what somewhat the big picture of emotion regulation processing in healthy controls. Maybe a few of you know this. And what happens in bipolar disorder? So I will try to summarize like 200 studies in one slide. So this is not a perfect summary, but I will speak mostly about studies in patients out of a major episode in the euthymic phase, just because for those who have seen many patients, they have psychomotor activation, disinhibition, things like this. You cannot put them in a scanner for 1 hour. You may try, but and when you see studies about many patients in the MRI, you have to. I think there are two possibilities. Either they are not true bipolar patients. They score high for whatever reason, but they do not have the diagnosis is not good. Or else they get what we call hypomania, or they are at the end of their episode. So this is not real MRI of mania. It will be hard to do this. I won't also speak too much about what's happening in bipolar depression, because it's always hard to disentangle what relates to the trait of being bipolar and the state of being depressed, except if you are doing longitudinal studies, rescanning those patients, but many of you know that this is not an easy thing to be done. So there are not so many studies like this. I will mostly focus on euthymic bipolar patients, so trait-related things, we hope. And what the classical tasks that have been done in these subjects are like really basic emotion processing, face processing mostly. You ask people to recognize, classify emotions, things like this. This is the most common kind of study being done. And what we can see in those studies is hyperactivation depends on the studies, depend for the location, but mostly hyperactivation of this automatic emotion network, most frequently with the amygdala overactivation. Decreased activation of the prefrontal dorsal cognitive networks, and inability of the prefrontal cortex to modulate, to decrease, to downregulate the activity of the amygdala. And then the amygdala is chronically overactive in these subjects. And we think, this is speculative, we don't have a proof of this, that this is linked to the emotional dysregulation those patients have out of an episode. This is not in the DSM, but unmedicated bipolar patients out of an episode often describe what you can call emotional hyperreactivity, like the threshold of the emotional, to have an emotional response for them is lower than in healthy controls, and the intensity of the emotional response is higher. Just a classic example is that patients are telling you that they are at a movie theater, they are looking at a sad scene, this is sad, everyone agrees, but they are the only ones in the room to cry. This is something quite classical, and this is out of an episode. So, and we think this may have something to do with this overreactivity of the amygdala-related networks. And I told you also that there was some kind of inability of the prefrontal cortex to downregulate the activity of the amygdala. This can be seen using fMRI or DTI-based tractography, and it's easy there to find that the structural integrity of the uncinate fasciculus is specifically altered in these subjects compared with the other tracts, and functional connectivity also is impaired in these patients. I won't go into the details of increased, decreased, it's really highly variable, you know, it can be misfits. And then there are some models now thinking about these frontolimbic networks as the core dimension of bipolar disorder, at least from MRI point of view. And we try to do these kind of studies, like assessing if early trauma, which is the major environmental risk factor for bipolar disorder, affected this network even in healthy controls. And now, from many studies, we know that this is the case. But we also tried to see if a very specific treatment of bipolar disorder was able to induce some plasticity of this network. So we conducted a randomized clinical trial of a psychotherapy, which is called emotional psychoeducation, which was developed in Barcelona by Edouard Vieta and people like this, which aims to focus really on the emotional regulation in patients with bipolar disorder. So we did 2 arms, one with this is a group therapy, one with classic support therapy for bipolar disorder. We scanned patients before and after, and we saw that only in the group with the emotional psychoeducation, we could find changes mostly in the prefrontal limbic functional connectivity. So it looks like that this may be considered as a dimension for bipolar disorder. Of course, all these studies have what we now call the classical statistical issues, mainly about small sample sizes, power issues, things like this. And I won't discuss this too much, but I think the main thing for bipolar disorder, having small sample sizes, is that you consider bipolar disorder as a uniform, homogeneous disease, and we do not know if this is the case. But you cannot compare subgroups of patients if you have 16 patients or 20 patients. So we went to multi-centric studies. We were very lucky, but we were very lucky at this time. It was in 2012 because we had 3 sites: one in Paris, one in Mannheim in Germany, and one in Pittsburgh. This is where the physicists were very useful. In fact, we were lucky because we had the same scanner, the same head coils, and then the physicists at Neurospin coded the binary of the sequence for T1 and diffusion and spread it out on the 3 sites. We wanted to decrease the inter-site variability. I will show you the result at the end about the interscanner variability. So we conducted, won't go too much into the details, this is what I put about tractography based on this 3-site study. And what we found was not what we expected to find, of course. We found these uncinate fasciculus changes with, using fractional anisotropy, but the largest effect size was on the corpus callosum. And then we opened a handbook about the corpus callosum and its role in emotional regulation. And we do not know much about the role of corpus callosum in emotional processing. Even lateralization of emotion regulation, emotion processing is something highly debated, with some people saying there are really many things. It's not like, I don't know, reading, of course, things like this, or motor skills, things like that. The lateralization of these processes is something really unclear. So we didn't know what to do with this kind of results, but the interesting thing is that we had a large bunch of patients. At this time, what was a large bunch of patients with bipolar disorder, 120, something like this, and we were able to look for subgroups of patients to see if subgroups of patients were associated with specific anatomical features. And we tested several things, and the only relevant subgroup we found was half of the patients had delusions or hallucinations during their episodes, which is called psychotic bipolar disorder, and half did not. And all the results were driven by those who had psychotic bipolar disorder. And then it relates also to some genetic literature phenotypics saying that psychotic bipolar disorder may be a very homogeneous subgroup of bipolar disorder with a specific biological framework. So we continued to grow with larger sample sizes. Thanks to the Enigma collaboration, for example, we are driving this subgroup, which is a bipolar disorder diffusion working group. Now we've got like 3,000 subjects, but we still have to be careful because, for example, in the previous study, we looked also at the side effects, despite having the same scanner, the same head coach, the same software to launch the sequences, etc., the same sequences. Side effects were of the same magnitude as disease effects. So, and we have the same, really the same setup, which is not the case in these now very large collaborations. So we've got to be careful. We thought about it. This is hard to prove, but then we discussed about this, and then maybe just patients are different. Even if this is bipolar disorder with the same diagnostic criteria. For example, me, I was working in a deprived suburb of Paris, mostly with inpatients, so probably severe patients with bipolar disorder. My colleague in Germany was having an outpatient clinic in a very rich region of Germany, and Mary Phillips in Pittsburgh was recruiting her patients through advertisements in the newspapers. So maybe those three strains of patients have something different. This is hard to prove, but we think that they are different here. So we have to be careful with those very large data sets for which we do not have so much insight about where the patients are coming from, etc. Anyway, we tried to analyze these huge data sets. We mostly found exactly the same kind of results as before, with changes in, for example, in white matter, anisotropy, mostly on the anterior part of the brain, not on the posterior region, so it's something quite specific, not like in schizophrenia where you find things all over the brain. And with the largest effect sizes, again, in the corpus callosum. We tried to do some classification just to see if it was possible to classify patients from controls using MRI, just to train ourselves. And you know, I'm at Neurospin. There are, for those who know this, there are maybe the major the inventors of the scikit-learn library. You know, it's I think the most used library in Python for machine learning. So they are very strict on cross validations, really strict. And then we ended up with figures like now maybe with 3,000 with 65% of accurate classification. This is above chance, but not so much. You know, I think you can take an 80-year-old boy discussing 5 minutes with the patient. This will be, he will be better than. Yeah, this is the case. So we just have to think that there's some signal in the images, but I'm not sure that using this at some point to do diagnosis would be efficient. Just to put this. Anyway, we also try to do unsupervised clustering based on the images to see after this if we could identify relevant subgroups with different, I don't know, clinical demographic characteristics. So we did this. This is also not easy because if you take images, ask the computer to do subgroups, the computer will really very well discriminate men and women, smokers and non-smokers. It works very well, things like this. So they developed at Neurospin something to identify those sources of variability in the healthy controls first, remove it somewhat from the patients, and then see what specific variability in patients. And we ended up only with 2 groups, those red and green dots, and we compared them for all their clinical characteristics. And the only difference between them was the intake of lithium. And lithium is the main, I didn't speak about, but it is the main first-line treatment for bipolar disorder. We do not know how it works because it was discovered, in fact, here in Melbourne, but by chance. The John Cade, who discovered it here, read really all the German papers of doctors working on gout. Maybe you know this is a rheumatismal thing. And this gout is related to calcium depots, and this is also a cyclic condition. So these German doctors thought that it has something to do with bipolar disorder, which is cyclic. It has nothing to do. This was a mistake, but this is interesting. And then they thought that lithium, which is a salt, may remove calcium depots, and this doesn't work too. But anyway, those German doctors noted that it seemed to change something in the mood of our subjects. And then they backed it. And 50 years ago, John Cade used this to treat 10 patients with mania, and it worked. And 2 died because, but it worked. But so this was discovered mostly by serendipity, you know, by errors at the start, gout, nothing to do with bipolar. And then we do not know how it works because of this. This was not designed using computers. So this was something interesting, and this led us to a large bunch of study. I will go just to understand how lithium works, because if we understand how lithium works, maybe we can understand something about bipolar. One go. Once we have those neural networks with bipolar disorder, with overactivity of the amygdala, etc., what can we do with this? So I think there are 2 things. We tried first to do like new treatments, investigational treatments of bipolar disorder in subjects out of an episode, but still with some symptoms. At Neurospin, we implemented, we did a real-time fMRI neurofeedback setup, and this is also. I know that there's one on the 7 Tesla here, but this is really not easy because to be able to have in almost real time the fMRI, the bold level of the amygdala, for example, to display it to a subject. You have to get access to the raw images in real time, and then you can have access to this, but then each time, for example, the software version of the MR platform changes, you have to redo most of the work, and it changes every time. So anyway, the thing was to recruit patients with some depressive and anxious episode, doing some emotion regulation tasks I will show you, and display them after a few seconds, the activity of the amygdala, in fact, the ratio between the activity and the amygdala and the prefrontal activity. And the goal for them is to increase, to decrease the activity of the amygdala here. You can see those kind of papers in the literature, not in bipolar, but in other settings. It's not, I don't know if someone has tried to do some fMRI or EEG neurofeedback by him or herself, but it's really hard to like control the activity of one brain region. We are not tailored to do this. Even I had to just control the activity of my supplementary motor region within the scanner, and really it's tough. So in the literature to do this, most of the studies say it's better not to give any instructions to the subject, and the subject will find his or her personalized way to do this. We couldn't do this, so we trained subjects first with a few strategies, which were some bits of mindfulness, some bits of cognitive reappraisal, and also, if we know it doesn't work much, but distraction. And then, after they learned this, they could choose their preferred strategy, because we tried to do this with pilot subjects without any instructions, and really they couldn't do this. So this is a study that I left. This is we have now 10 subjects. Our goal is to have 64. This is really a RCT, randomized clinical trial of 3 sites in France. The control, this is not easy to find a control to do this. The control intervention is real-time fMRI neurofeedback, but of a region supposed not to have anything to do with bipolar disorder, so something really posterior. And then we have 3 sessions of real-time fMRI neurofeedback. I will try to show you. So this is a setting at the console, MR console, and so you see here these are neutral images, and you see here the activity of the amygdala with a small, but this is decreasing, and then these are negative images, and you can see the activity of the amygdala in real time increasing, and then this is the feedback to the subject. We summarize 5 or 6 repetitions to the subject, and then neutral images, and the activity of the amygdala decrease, etc. And the instruction for the subject is to decrease the activity of the amygdala and or increase the activity of the prefrontal cortex. We will see if it works. We will see. So this is one of the line of research where we are using what we have learned using neuroimaging. Another thing we go briefly about this is studying really the effect of lithium using neuroimaging. So there are many studies till 2000 showing that in longitudinal studies, if you scan patients with bipolar disorder before and after the intake of lithium, you can see whole brain gray matter increase. And this goes very fast. Like in 2000, I think they gave it for 4 weeks to 10 subjects, and they see like a 5% increase, something. But this is replicated. We did cross-sectional studies using this search, and we found the same, and many longitudinal studies also have replicated this effect. I think we still have to be cautious because gray matter volume is a computer reconstruction of some radio frequency signal, created signal in the brain. We have to be cautious, but some preclinical studies suggest something similar. So really, this is, I don't know if it will work here, but I was at the coffee machine. There's no coffee machine, not so many coffee machines here, but at the Atomic Energy Commission, and I spoke about this, and the physics guy said, oh, you know, you can do MR of the lithium. Well, we don't know, but so we are doing, basically, we are doing MR of the proton, of water proton, but there's no clear obstacle to perform MR of other things. So nowadays, MR of the sodium, phosphorus are quite popular. Not easy to be done because of the lower concentrations of those components in the brain. For lithium, we do not have any lithium occurring naturally. And in patients with bipolar disorder taking lithium, the concentration of lithium is 100,000 times less than the water concentration. So we are using centimetric cells with a 7 Tesla scanner, with a specific dedicated coil, because the frequencies, resonant frequencies are not the same. Anyway, they did this. First, we validated this in rats. We did this in rats, sacrificed the rats, assessed lithium concentrations in the brain, and compared it with our MR signal, and it worked. So we did it in patients. And here it's from one patient, but you can see that first the distribution of lithium in the brain is quite heterogeneous, which is not something obvious because we do not have lithium pumps. There's no lithium in the brain, so we do not have lithium pumps, lithium channels. So this is something that we've seen. And also what we've seen, we wanted to see where does the lithium goes most, and what we found, so this was statistically significant only in the left, but in fact this is both-sided. And there's an accumulation of lithium in the hippocampus. And then some people started to say, you find that lithium goes in the hippocampus. People found that found gray matter increases with lithium. This is easy. This is the only place where new neurons are born in adults. So these must be new neurons. We do not think this, but to go further, how can we do? So what we did was using new techniques of MRI. Same in this neurospin, they are doing this. I think in psychiatry we've got, in bipolar disorder and in psychiatry in general, we've got two main issues hampering any progress. First, we do not have animal models of our disease. We have animal models, but with all the limitations that you may imagine. The only animal model that I found really convincing in some kind of psychiatric condition is in autism. Maybe you can see these videos of non-human primates with a gene defect MECP2, and really, but they look having autism in the videos. Anyway, this is not very, on an ethical point of view, the engineering monkeys to make them autistic. So this is done in China. And really, it's a weird thing. So, but except from this, depressed mice. I don't know if manic. So you can do manic mice. So these are mice running all the time. You can do depressed mice or rats. These are mice not struggling, let's say. But even now, we do not have, for example, rodents cycling, having excitation and then depression. And of course, these excitation and depression have huge limits, of course. So I think this is one thing. We do not have animal models, and we do not have histology. If you are, I said this to liver physicians and things like this, but it's easy to study liver conditions. If you've got something in the liver, you just take a bit and look at in the microscope, and then it's easy. In the brain, it's harder, and in psychiatry, it's even harder than in neurology, because the typical age of onset is at 15 to 25. So you can have the brains of the patients if they die at 50, 60, 70, but it won't be very relevant after 40 years of episodes, lithium, alcohol, I don't know what. And so we will never have this, I think, or with very few samples. So we are using some new MRI techniques. I won't go into the details, but that are trying to assess with MRI. They are building physical models of MR and trying to assess myelin, synaptic contents, density of fibers, diameters of cells or fibers. We had the chance to work with someone who developed one of these in gray matter to study number of synapses. Just be careful with number of synapses. So we did this in patients in 2 sites. This is a multi-shell diffusion imaging. And what we found in 3 sites, in fact, in Paris, Grenoble, and in Canada, was that patients taking lithium had a higher index of this synaptic content in the prefrontal cortex than patients without. We have to be careful because there's much physics work behind this, with many assumptions. But this led us to think that in 4 weeks you can have, but it was not obvious for us to have new neurons with lithium, but mostly we think synaptic branching or synaptic anatomical changes, things like this. So to study this is the last study I left in Paris. We tried to do really like what everyone calls like translational studies, but There is now a PET marker of the synaptic content, which is an antipsychotic, an anticonvulsant, which goes on every excitatory vesicle. So it counts, really counts the number of synapses. So we will do this in patients with bipolar disorder before and after lithium. We will do also at the same time, because we have a PET MR scanner, the same MR thing that we did in the previous study to see if it really matches. And we will do this in a bipolar rat. So I've said that bipolar rats before and after lithium. But the good thing with the rats is that we will sacrifice them and really count the number of synapses. And people doing this also explain me that they could now assess the anatomy of the synapses. You have different kinds of synapses that they can count to really better understand if we get a change in synaptic density, what's the histological counterpart of it. So for the last 5-10 minutes, I would like to go into what I would like to do in the future here in bipolar disorder. I think after having taken part in those studies, even the Enigma studies where we end up with 3,000, 4,000 subjects, that these kind of studies have reached a point where I don't think having more subjects or fancier MR techniques will help us much, because we are doing mostly snapshots of the brain, captions of the brain, and then we try to explain things with this, and we have models, but really, this was also because I was at the Physics Institute. I could see that they didn't work. Physicists are not working like us, and they often come to me and say, "Oh no, you are not doing science." So, yeah, but I think they are somewhat right. They say, "Okay, we are building models of how a particle should behave." I understand it. And then we are designing an experiment. We are testing our model. Our model stands or fails, or we refine the model, and then we have a new model that makes predictions, and then we iterate this. And in bipolar disorder, I think maybe in autism, some people are doing things like this. There are some broad hypotheses of why there's autism, etc., excitatory, inhibitory things. In bipolar disorder, we do not have. We just take captions and draw inferences from this. So maybe it's, I'm not bipolar, but maybe I had some kind of manic episode, but I wanted to draw a model, to build a model, something similar, and test it, and have a model that would explain everything, bipolar disorder, and would have the ability to do predictions. So this is why it take 10, 15 years to check. And maybe because I am from France, I started from a very famous and used concept in biology and medicine, but not in the brain. So this is Claude Bernard, a famous French physiologist, and he wrote the concept of some of you working on the sleep studies know this concept very well, homeostasis. Homeostasis is basically that many of your variables in the body are tightly regulated despite external perturbations to stay within certain limits that will optimize your rate of survival. The most common, one easy way to think about it is about glucose level. So glucose level has a certain, in the blood has a certain range. You can have a large cake, eat all the cake, so the glucose level will increase, but there will be something that will bring back, despite this huge perturbation, your glucose level to an optimal level. And if it doesn't go to the optimal level, if you are not successful to do this, it's because you have some disease, diabetes, and you will have detrimental effects of this. And it doesn't go also too low, because else you will faint, etc. And it works for many functions in the body, blood pressure, electrolytes content. In the brain, it has not been studied so much, except for circadian studies. This is quite an obvious phenomenon. This is called sleep homeostasis or pressure sleep. If you do not sleep for one night, the night after you will have an increased what you call pressure of sleep. It will be easier for you to fall asleep, and you may sleep longer and in a more efficient way. But except this has not been studied much. How does it work, homeostasis, like for glucose? But this works for everything. This may be modeled really easily. You have somewhere in the brain, somewhere in the body, a sensor which monitors your glucose level. And then in this sensor, you have an error detector. So if it goes above or below some limits, you will have an error signal. And then at the end, you will have an effector. And for glucose, it will be liberation of insulin that will bring your glucose level back. And then there's a loop like this. I don't want to go into too much complicated models. That's good for me. So it's a very simple thing. Does it work for brain functions? So I think for sleep, and I haven't gone into details, but circadian rhythms are highly disturbed in patients with bipolar disorder. For example, when they start a manic episode, they sleep less and less, and they don't have this increased pressure of sleep. They sleep like 1 hour less, 2 hours less before starting a full-blown episode, and they do not feel tired, etc., till not sleeping at all for some patients. And this is so strong that we tell patients, if you sleep less without any reason and you still feel full of energy, please just give us a phone call or take this medication even before calling us, because this may be start of an episode. So they have some, I think this is a model, some kind of dysregulation of their homeostasis in the circadian functions, but even, for example, at other scales or other levels, mood, we do not think about mood as something homeostatically controlled, but during COVID in UK, because people were locked down, they were happy to take part in some studies, and at Oxford they conducted a study in 100,000 subjects doing EMA, ecological momentary assessment. They were asking two things to subjects: what are you doing now, and what's your mood level? This is in the general population. And what they found was that when in the general population their mood was slightly increasing, people were more prone to engage into housekeeping, which is supposed to be a boring activity decreasing your mood level. And when their mood level was slightly decreasing, they were more prone to engage into physical activity, which we know, we try to do this with patients, increase mood. So they tried they ended up using modeling mood as a homeostatic, self-regulated variable, and this doesn't work in patients with bipolar disorder. When their mood starts to increase, it will increase more and more without any control, and the same when it goes down. And we even tell them, you should do that activity, and for many reasons, for motivation, we cannot do this. So I think this may be also another manifestation of altered homeostasis in the brain of patients with bipolar disorder. There are some other examples. I haven't spoken about it, but another line of treatment of bipolar disorder, also discovered in weird ways, is anticonvulsants. And the job of anticonvulsants is basically to stabilize membranes and synapses that are known to be dysregulated because of some alterations in the calcium metabolism in patients with bipolar disorder. So I will try to test this. This is not easy to operationalize, but in fact the whole model the whole assumption is that patients in many dimensions are more liable to external perturbations and will have difficulties to bring back some brain variables to the usual range. So we put a grant proposal for this with, for example, Elise about a marker of their sensitivity to light. She has in her studies here found that patients with bipolar disorder had a higher pupillary response to blue light than depressed patients or than controls. And this may suggest also because that patients are more liable to perturbations in their circadian rhythms. And it's something clinically relevant because when you've got a patient who will do an intercontinental flight, you warn him or her that he or she may relapse because of this, of the time shift, of this external perturbation. I will try also to do some actigraphy measures, but we will need some long-run actigraphy measures to see how the subjects are regulating their sleep and also their mood level using some software, CIMA, ecological momentary assessment developed here. So I will try to assess the model on several scales, even doing electrophysiological studies at the Florey Institute in rats model of bipolar disorder. And that's all. Thanks.
Thank you, Josselin. As a former physicist, I really enjoyed your comments. So let's have questions from the students first.
Curious question. First of all, thank you for the lecture. It was really engaging and interesting. And what made you come to Melbourne?
Wait. No, it's more a matter of personal things. Just because thinking that I don't know if it's an advice or a good or bad advice. I will tell you in 5 years if I'm happy or not with this. Just that after like 15 years running the same lab, I found this very good just to switch, to put some pressure, not put some pressure. I don't know if it's the same in English, leaving your comfort zone.
Yeah, we say that.
And because you know when you're running a lab for 10 or 15 minutes, I was a professor at Paris, and it was easy to get funds because people were coming to me saying, you know how to do this, can you do this for me? And I just put my name and I had some money to do this. Students were, I don't say it was easy, but I was in the Institute of Stanislas Dehaene, so the same people were coming to Stanislas Dehaene saying, do you know someone, a psychiatrist? So it was like easy. And at some point I realized that I was becoming lazy. You're not lazy, you know, but just to have new ideas. For example, this idea just came because I said, I'm going to Melbourne. Will I do exactly the same? But no, I think just doing a move like this may force you to think again about what you're doing, get new ideas, and maybe probably this will be physicists have always failed models. But still, I think it's good to do at some points of your career doing this.
Amazing that it's easy to get funding in France. I think we're all going to be crazy now.
Easy, but first, I'm not sure here, but in continental Europe, I really found that it's not a good thing, but when you're a medical data, it's easier because there are not so many others, so the institutions wanted to find I don't know, at some point when you know everyone, and you know how the system works.
It gets boring. Maybe.
No, but easier and not challenging enough.
Yeah.
Maybe not, for me, I reached a point it was not challenging enough. Really, I had some, because we had, for example, at this time, the only 7 Tesla in Paris, and there were only 2 in France. Some psychiatrists wanted to study, I don't know, circadian networks, and they realized that they had to go to a 7% like this, but they had no imaging background. So automatically they were coming to me, and they were designing the grants and doing everything. Sometimes it worked, sometimes it didn't work, but it was not so much work for me. it's really a question. It was not so much work for me. Now doing an ideas grant from scratch, but it forced me to really to think about, and even I don't know if you know these enigma studies, but I'm co-leader of one work package, and it's maybe this is a good thing, and the guy who did Paul Thompson, he was right doing all these things, but at some point it's not challenging enough. You know, you just collect like 3,000 things. You do, even if you do, I don't think something like univariate mass statistics, you end up with some nice results. You send this to journals, and they take it because you have 3,000 subjects, even if it's not very interesting. And I don't want to say my was not interesting, but we learned a few things, but it's not. Yeah, and I liked what the physicists were doing. This was much more challenging, having to think about it. But to do this, you had to sometimes to switch and to move, to leave things, because also the bad point was that I was really struggling with time, because psychiatrists or neurologists were coming to me like every week. Oh, I want to do this, I want to do this, can you do this? And then I had no time to think about other new things, because there was, I had to say yes to still stay in the easily funded things.
Very good insights. Other questions? Yeah?
Very interesting. How do you feel the corpus callosum results with your homeostasis? Do you think it's any, like, because it's a big track connecting the two hemispheres, do you think it plays a role in that sort of model of theory?
First, it was really puzzling to me that we didn't know so much about the role of corpus callosum, except in visual field. The visual field, you know, there are like divided studies of divided visual field, but it's easier to understand. And the same with language processing, because it's really highly virtualized. We don't know so much about this. This will be hard to study, because you've got subjects, for example, with agenesis of corpus callosum, but because it's mostly congenital, people are adaptive. I even saw, I was scanning many patients, many controls for other studies, and from time to time we had subjects with incidental agenesis corpus callosum, and I had to assess the subjects. Really, sometimes nothing. They have no corpus callosum, and they were like 100% normal. Yeah. So really, I don't know. There's one technical trick, which is also that assessing the fibers in the corpus callosum is easier, because all your fibers are packed and have the same orientation, which is not the case in the, maybe it's related also to this, DTI being more efficient here, DTI modeling being more efficient here. But we don't know.
Yeah, I had the same thought, where the corpus callosum just simply has greater SNR, so it's easier to have an effect.
Yeah.
But I also noticed that in schizophrenia, the corpus callosum is